Okay, so how do our cells know when to divide and when to stop dividing? Well, it turns out that our cells have molecules, proteins, embedded in their cell membrane. The cell membrane is the determination between what's inside and outside the cell. So it's essentially having a receptor located at the entrance of the cell. Those receptors can receive chemical signals that our bodies send to the cells to tell them what to do. When the receptors receive these chemical signals, they send this signal to molecules inside the cell. Those molecules inside the cell then continue to send signals to other molecules until the information is received by the nucleus. I like to think of this as the game of telephone. If you've ever played the game of telephone or you know how it works, one person whispers into the ear of a second person, then that person sends the same information whispered into the ear of the third person, and so on. Eventually, the information is received at the end of the line. Of course, if you've ever played this game of telephone, you may have played with someone who unintentionally or intentionally messes up the information. If you ever had somebody intentionally mess up the information, this is sort of like a mutation in a cancer cell. If there's a mutation in any of the proteins along the way that send these signals, then the signal information won't be sent properly. In fact, some of these proteins could be sending signals that the cell needs to divide, even when no signal was sent by the body. So the cell's nucleus receives information that it will need to divide soon or needs to divide now, while the body has not sent any signal indicating that. That can cause the entire cell to undergo the cell division process in an uncontrolled way. That leads to cancer. This whole process is so complex, it involves many, many proteins. You need to have a mutation in the cell cycle checkpoints between G1 and S, a mutation in the cell cycle checkpoint between G2 and M, and then also another mutation in a cell cycle checkpoint in the middle of the mitotic phase. So in fact, there are 3 cell cycle checkpoints in this process. But in addition to that, you also have what I sort of consider as bouncer genes. These are genes that make proteins that check to make sure everything in the cell is functioning properly. It's as if you have someone keeping an eye on the entire system to make sure it's going well. And if it does not, those proteins send a signal to the cell that the cell needs to go through apoptosis, which is cell suicide. Those bouncer proteins also need to be mutated and not functioning properly in order for cancer to develop. So in order to form cancer, we need to not only have mutations in the 3 cell cycle checkpoints, but also in the tumor suppressor genes, those bouncer proteins I was talking about earlier, and in a number of other instances as well. So generally, malignant tumors that have eventually metastasized will involve mutations in many pathways. First, the cell cycle checkpoints and the tumor suppressor genes need to be mutated to keep the cell dividing uncontrollably. But that's not the only thing that needs to happen in order to form a tumor. The cell must then have a mutation, allowing it to take in more food than usual. This allows the cell to grow more rapidly than its neighboring cells. In order to metastasize, the cell must then synthesize a molecule known as matrix metalloprotease. The matrix metalloprotease is a protein that allows the cells to digest tissue connections and free itself from the neighboring cells. The cancer cells can then tear through tissue walls and move around the body. To move to a new area of the body, the cancer cells must then mutate even further to dissolve into blood vessels and move through the bloodstream. Further mutations need to allow the cell to survive while it's moving in the bloodstream. and then even more mutations to allow it to attach or adhere to a new location in the body. And once there, the cell has to have further mutations that encourage new blood vessel growth so it can grow into a new tumor in the new location. This is why we say that cancer is a result of an accumulation of mutations, because of all of the mutations required to get this to occur. Remember, too, that all of the genes in our bodies come in pairs. Some genes are dominant and some genes are recessive. These mutations that are dominant mutations require only one mutation in order to generate a cancerous trait, whereas recessive mutations need to have both copies of the genes to be mutated in order to have the cancerous trait. Some individuals are born with a predisposition for cancer. This was studied in two commonly known cancer genes, retinoblastoma and the BRCA1 and BRCA2 genes. The retinoblastoma mutation is a recessive mutation, and it's inherited from parent to offspring. Usually, a child will receive one mutated copy of the retinoblastoma gene. It's called retinoblastoma because if the other copy of the gene gets mutated at any point, it can lead to cancer of the eye or the retina. If an individual inherits one copy that's been mutated already from the parent, they only need one additional copy in order to form the cancerous trait. That is why they have a much greater risk of developing cancer of the eye than someone without the mutation. It's not a guarantee that they will form cancer of the eye, but they do have a much larger chance. The same is true of the BRCA1 and BRCA2 genes. BRCA stands for breast cancer, and there are two genes associated with breast cancer. These are genes that, when mutated, can lead to breast cancer. They're also inherited from family members, but just as a predisposition. If you inherit a single mutated copy from a family member, there is no guarantee that you will develop breast cancer in your lifetime. That means that these mutations are also recessive, and all you need is to have the other non-mutated version get mutated in order to form the breast cancer trait. Some individuals can inherit an 85% risk of developing breast or ovarian cancer in their life. And they would know this because they would have their mother's sisters or aunts on their mother's side, possibly their sisters and their grandmothers, developing breast and or ovarian cancer earlier on in life, around the age of 40. But again, an 85% risk is not a guarantee. It's just a very likely chance. Now, these two examples of genes mutated in cancer, while they've been widely studied, are in fact very rare. So most individuals that acquire breast cancer in her lifetime are not inheriting the BRCA1 or BRCA2 genes. It is completely random that they acquired breast cancer in their lifetime. I cannot emphasize that enough, that if you know anyone who's ever had breast cancer, it is very, very likely that their development of breast cancer was totally random and unavoidable, and that they do not have breast cancer in their family line. The only time that it would be of concern is if you have a family tree much like this, where you have multiple family members that have died of breast cancer early on in life. When scientists looked to see what kinds of mutations are most common in cancer, they fell upon the mutation in a gene known as p53. p53 is the most common gene mutated in cancerous cells of all types, prostate, lung, colon, breast, and many other types of cancer. Despite the fact that it is the most common gene mutated in cancerous cells, it's still only found mutated in half of all cancers. What this indicates is that when we say cancer is an accumulation of mutations, the mutations that lead to cancer are different in every patient. They are unpredictable, and nothing that you can assess or determine can lead to cancer. P53 is one of those tumor suppressor genes that we talked about. When functioning, it keeps cells of the body from growing out of control. If it is mutated, though, it loses its ability to keep cells from growing out of control, and it cannot stop uncontrolled cell division. This does not mean that you instantly develop cancer, but what it does mean is that you've lost the ability to regulate the formation of cancer. Other genes can be mutated that lead to cancer are referred to as proto-oncogenes. Proto, like prototype, means that they are genes that are not quite oncogenes, but they could become oncogenes. Oncogenes are like oncology. Onco meaning cancer, and gene meaning gene. So these are genes in your body that could develop cancer if they are mutated. The proto-oncogenes are the types of genes that are those cell signaling genes involved in the cell cycle checkpoints. And when they are mutated, they result in a gain of function over signaling to the cell, leading to cancer. Proto-oncogenes are likely to be dominant mutations because you only need one copy to send a faulty signal, whereas tumor suppressor genes are more likely to be recessive mutations because to lose the function of P53, you would need to lose the function of both copies of the gene. Now, how do we acquire all of these mutations that could result in cancer? We're going to be talking about that and cures for cancer in our next segment.